Okay, now please allow me to introduce our next presenter. So Dr. Koen van der Plas graduated in 2004 as Industrial Engineering Chemistry at the University of Ghent in Belgium. And before joining Nelson Labs, he worked for 2 years in different analytical/environmental labs, specialized in routine water and soil analysis, gaining great practical experience with various analytical techniques such as GC/MS, LC/MS, and ICP. So Koen started his career at Nelson Labs in 2006 in the GC/MS department of the analytical lab. And after a period of 7 years where he witnessed the beginning of extractables and leachables testing, he moved to the study director department where he worked for 5 years in different SD groups specialized and responsible for E&L testing of various pharmaceutical applications for the industry. In the last 5 years, he is part of the SD group at Nelson Labs, focusing on E&L testing of medical devices. And with that, I'd like to hand over to our next presenter. So welcome, Koen.
Okay, thank you, Marcus, for the introduction, and thank you, Ellen, for your presentation. So good day, everyone. Today, I will give a presentation on medical device, chemical characterization, what the toxicologist needs to know from the chemist. And my presentation will actually highlight how you come to the chemical data in the first place a toxicologist can do his or her risk assessment on. So here you can see a summary of my presentation. So I will first start with a short introduction. Second, I will describe the setup of a chemical characterization or extractable study in detail, from the selection of extraction solvent and conditions to the detection, identification, and quantification of the detected extractables. Third, I will also describe which possible issues a toxicologist might be faced with during a chemical characterizations assessment and how these can be addressed. And then fourth, I will finish my presentation with a conclusion, some takeaway messages. So let's start with the first. So chemical characterization, what is it? So in the ISO 10993-18 standard, so chemical characterization is described as the process of obtaining chemical information on your medical device, either by, and I indicated this in the left, information gathering, by performing a literature review on all information available for your device, or by, and this is indicated in the right, information generation, by performing chemical testing. So, and this is often a misunderstanding. Chemical characterization doesn't necessarily imply testing. Now, from the literature review of your device, the configuration, the material composition, the clinical use, etc., it can already be concluded that the device is safe or not. Now, if not, or in case there's insufficient data, you need to move to the right. And in that case, information needs to be generated by means of an extractables and or a leachables study. Now, extractables or leachables testing of a medical device, what is it? So in an extractables or leachables study, you are going to examine which compounds might migrate out of the device and can potentially enter the patient. Now, such an extractables or leachables study is often followed by a toxicological risk assessment to see which of these compounds are safe or unsafe to the patient. Now, related to this, it is important to highlight the difference between an extractables and a leachables study. So, in case of an extractable study, you are going to look for the substances that are released from the medical device using laboratory extraction conditions and vehicles. So, in this case, you are using severe conditions that exaggerate the real use. Now, in case of leachables testing, you are going to look for the substances that are released from the medical device during its clinical use. So here you are using less severe conditions. Now, however, seeing the use of some medical devices, this is not always feasible. So therefore, here it is better to speak of a simulated use study, where you're going to simulate the clinical use of your medical device as good as possible. Now, and herewith, you always need to keep in mind that your leachables will always be a subset of your extractables. Now, if a chemical characterization study or an extractable study is necessary, how do you need to set up such a study? Well, a first important parameter is the extraction. So ISO 10993-18 says to always use a minimum of 2 solvents of differing polarities. Now, we mostly use water for polar and hexane for non-polar conditions. Now, most authorities request a third semi-polar solvent for long-term implants or blood contacting devices. So in such a case, as you can see here, isopropanol is often used to mimic these blood contact or semi-polar conditions. Now, some remarks. By choosing the extraction solvents, you always have to consider the use. Now, in case, for example, there are interactions with specific solutions or drug products, additional extraction solutions might be needed. For example, if you are performing a chemical characterization study on a vaginal ring, you might need to consider also a water with low pH solvent to be included to mimic the acidic environment of the vagina. And for medical devices with indirect contact, like for example, infusion bags or insulin delivery devices, it can be appropriate to use a single extraction solvent that replicates the expected contacting fluids. But in any case, the choice of these extraction vehicles must be justified. And then lastly, you need to avoid solvents, as can be seen here, that cause huge swelling and or degradation, as you will obtain in that case an extractable profile that is not representative for patient or user exposure. Now, which ratios do we use? So therefore, we base ourselves on table 1 of ISO 10993-12. So as you can see here from the table, the 3 or 6 square centimeters per ml are the mostly used extraction ratios. Now, however, sometimes the extraction ratio is not feasible and you have to deviate from it. So, for instance, when you have a big metallic implant, a substantial amount of solvent might be needed to completely submerge a test item in an appropriate extraction vessel. And in that case, you might need to deviate from the 3 square centimeters per ml and use a lower extraction ratio, which is okay, as this table of the 12 standard is just a recommendation. Now, which temperatures and times do we use? So therefore, we use table 2 of the 18 standard. And you can see here, depending on contact category of the device, there's simulated use, exaggerated, and exhaustive extraction. Now, for limited contact devices, so less than 24 hours, simulated use conditions are recommended. Now, for prolonged, so more than 24 hours to 30 days, and long-term, more than 30 days, contact devices, exhaustive extraction is recommended. Now, exhaustivity is determined with gravimetric analysis, as you can see here. So non-volatile residue measurements on 24 hours extraction cycles are done. And if the non-volatile residue of the 4th extraction cycle, for example, here, is below 1/10 of the non-volatile residue of the 1st extraction cycle, then exhaustivity is reached. So in this example, you need to exhaustively extract for 96 hours. And then credible alternatives are applying exaggerated extraction conditions. Now, 50 degrees Celsius at 72 hours is an often applied exaggerated extraction condition. How do you need to detect your extractable? So let's say this is your medical device where you made an extract of containing your different compounds or substances that can potentially migrate out of the device. So you will have the small, the medium-sized, and the large molecules. And lastly, you will have the metals. Now we call these the volatile, semi-volatile, and non-volatile organic compounds and the metals. So the small molecules are analyzed with headspace GCMS. This is headspace gas chromatography with mass spectrometric detection. So typically here we screen for all kinds of monomer residues, solvent residues, residues from various polymer treatment steps, and small polymer breakdown products. The medium-sized molecules, the semi-volatiles, are analyzed with GCMS, gas chromatography with mass spectrometry detection. So typically here we screen for all kinds of lubricants, plasticizers, antioxidants, polymer degradation products, high boiling solvents, etc. And then last, the large molecules, the non-volatiles, are analyzed with liquid chromatography with mass spectrometry detection. So here your target compounds are fillers, plasticizers, antioxidants, anti-slip agents, etc. So, and by using these chromatographic techniques, you will end up with a chromatogram with peaks like this. Now, these peaks are representing the different compounds that migrate from your device, eluting at a certain retention time, as can be seen here, with a certain abundance. Now, the higher the peak or the abundance, the higher the concentration of your compounds. And then metals are analyzed with inductively coupled plasma with optical emission spectroscopy or mass spectrometry. Now here you can see the standard setup of a chemical characterization study. But of course, additional techniques can be added if this is deemed necessary. So for example, if you have a medical device and it is expected that your device will release certain anions, ion chromatography might be included as well. Now, at which threshold do you need to detect your extractables? So therefore, there's indeed the AET. Now, how is the AET defined? So the AET is defined as a threshold below which the analyst need not to identify or quantify leachables or extractables or report them for potential toxicological assessments. So if the AET is located here in your chromatogram, these 8 compounds need to be further followed up. Now, and here you can see the formula from the ISO 10993-18 standard. So the AET is expressed in micrograms per milliliter. It's a concentration-based threshold, and it is calculated from a dose-based threshold or threshold of toxicological concern. Now, to know which DBT or TTC you should use, there's an additional guideline, indeed, namely ISO 21726, and in this guideline there's a table specifying the applicable TTC based on the duration of body contact. Now, so you can see here, depending on the contact, you need to use 1.5, 10, 20, or 120 micrograms per day. Furthermore, A and B are coming from the used extraction conditions. So A is the number of devices used to generate the extract, and B is the volume of extract. And then there's C, that's the clinical exposure of the test item. And lastly, you have the uncertainty factor. So this is the uncertainty factor for the chromatographic screening method under evaluation. Now at Nelson Labs, we usually use an uncertainty factor of 2 for GCMS and 5 for LCMS. Now in the 18 guideline, it is explained how you can calculate this uncertainty factor for these chromatographic screening techniques. So a short example, let's assume that we have a catheter. So, and this can be used up to 33 days. Then we have a TTC of 120 micrograms per day. Let's say that we extract one catheter in 100 ml, then A is 1 and B is 100. And let's say that the clinical exposure is 1, so 1 test item can be used per patient per day. And for, you can see here then for GCMS, where we apply an uncertainty factor of 2, we will end up with an AET of 600 microgram per liter. Now, identification of extractables, this is done with mass spectrometry. So each peak above the AET represents a compound with a unique mass spectrum, as can be seen here in the left. So commercial libraries like NIST and Wiley, they contain mass spectra of many compounds, but often they do not contain the medical device-related compounds we are interested in, leading to sometimes incorrect identifications, as it is illustrated here. So with a commercial library, you will end up with a upper right component, this oxyrane-like compound, but actually it's the lower compound, the crown eater, which is not in a commercial library, but we have this compound in our in-house library. So, however, a correct identification is crucial, as this is the link to relevant toxicological information and correct patient exposure, and thus patient safety. So therefore, it is best to have an in-house library that is built on compounds detected in extractable studies of medical devices. Now, a good library or database is extensive, as the medical device world is extensive. There are a variety of process parameters, solvents, glues, sterilization, etc. And there are a variety of materials, metals, polymers, biodegradable materials, and so on. A good database should also contain relevant compounds. And a good database should also continuously grow, as constantly new materials are being invented. And a good database, of course, should be reliable. And last but not least, all compounds should have a clear identification status. And it is crucial that the toxicologist knows how certain the identification is. And here you can see the 4 identification levels that we use at Nelson Labs for the reporting of extractables and leachables. Now, these identification levels are done according to the classification levels as described in the USP 1663. Now, in case of an unknown, you don't know anything. The next identification is a TEC, a tentatively identified compound. So here you have a low hit rate with your library, so it's unlikely that this is your compound, but from the mass spectrometric information, you know a class of the molecule, or a molecule formula or composition. The third identification is an MPC, a most probable compound. So here you have a high hit rate with your library, and you know the structure. So you can give a name of the compound and ideally a CAS number. But the compound is not confirmed with an authentic reference standard. And then the fourth classification is identified compound. So there is the compound is 100% sure, as you have confirmed it with an authentic reference standard. So you can see here, by moving from the left to the right, there's an increasing amount of confirmatory information and an increasing certainty that the identification is correct. So, and of course, this is what it looks like for a toxicologist. Now, at Nelson Labs, we have our own database, the Nelson Labs Unique Compound Screener Database. Now, this database was started approximately 17 years ago based on the injection of analytical or synthetically synthesized standards, and it is still updated regularly with new standards or identification. So here you can see a little bit how it works. So for every compound standard, we record the retention time, the mass spectrum, and the relative response factor. And then in the end, extracts are screened using this library and the information therein in combination with the standard commercial libraries. So, and here you can see the latest update on the number of compounds that we have in our database of our library. So you can see here, we have more than 2,000 compounds for headspace GCMS, more than 10,000 for GCMS, and more than 3,000 for LCMS. Now, of course, you can imagine that this is a very powerful tool in extractable or leachable studies. And then the last step is, of course, the quantification of your detected and identified compounds. Now, here you can see the 3 definitions from the 18 guideline, listed from the most to the least accurate. So let's start with the first, the quantitative analysis. So in the ideal case, you have a calibration curve for each detected compound. However, this is not possible in a screening approach, as you do not know beforehand what to find. So, for example, it is not uncommon that for an extractable study of a polymeric device, you will end up with 50 or more extractables. Now, recording calibration curves for these 50 or more extractables is practically an impossible job. So the second best you can do, and this is the second definition, is the semi-quantitative analysis. So there you estimate the concentration based on the response of an internal standard, considering relative responses. So as you can see here in the picture in the right, it is possible that compounds present at the same concentration of the internal standard do not give the same response. And with a relative response factor, you can correct for this difference in response. Now, of course, this can only be done for identified compounds recorded in your library, of which you have these relative response factors calculated and listed. And then lastly, and this is the third definition, you have the estimated quantitative analysis. So there you estimate the concentration based on the response of an internal standard without considering relative responses, as can be seen here in the picture in the right. So you assume that every compound gives the same response when present at the same concentration. So in this case, if you can't do a relative response factor correction, you will end up with an estimated analysis and concentration. Now, screening methods are estimated or semi-quantitative. And so quantitative analysis, we also call this targeted analysis. So, and here you can see, again, by moving from the left to the right, you have an increasing certainty on quantity. And again, this is what it would look like for a toxicologist. Now, we consider the second quantification, the semi-quantitative concentration, sufficient for a toxicological risk assessment. So, and in the end, you will end up with a report with result tables like this, where you will have a combination of identified compounds, most probable compounds, tentatively identified compounds, and unknowns at a certain concentration. Now, which are possible issues a toxicologist might be faced with during a chemical characterizations assessment, and how can these be addressed? So, a first possible issue is indeed that the identification level is insufficient if there are too many unknowns. Now, in that case, we offer second pass testing. Now, in second pass testing is done with high-end equipment, as can be seen here. So in that case, the extracts are analyzed with GC-QTOF, time of flight, for the semi-volatiles, and LCQ exactive orbitrap for non-volatiles. So here you are using high-end mass spectrometers with very high resolution and high mass accuracy in combination with multiple ionization methods and tandem mass spectrometry. So with this tandem mass spectrometry, these MS2 data, there is the possibility to structurally elucidate the unknown or the compound of interest, as it is illustrated here in this example. So there are possibilities to further identify these unknowns. Now, which can be another issue? Well, the issue can also be that you end up with an extractable study with too many extractables at a too high concentration. Now, in that case, I remember my first slides, you can move to a leachable study, a simulated use study, where you simulate the real use conditions more. Now, the conditions of a leachable study are more case-to-case dependent, but the idea here is to simulate more the real use conditions. And you can do this by using, for example, a simulating solvent. So, for example, if you have contact with blood, you can use 40% ethanol in water instead of 100% isopropanol. When you have contact with wounds, you can use saline to mimic sweat in combination with 40% ethanol in water to mimic blood. If you have contact with certain drug products with certain pH, you can use water at that pH in combination with a binary mixture. So a certain percentage ethanol or isopropanol in water to mimic the organic content of your drug product. And next, second, you can also lower your temperature. So instead of using the 50 degrees Celsius, you can go for 40 degrees Celsius, if it's applicable, or 37 degrees Celsius, the body temperature, or 25 degrees Celsius, the room temperature. And lastly, you can also shorten the extraction time. So for example, when you have an intermittent urinary catheter, which is only used for 5 minutes per catheterization, it can be enough to reduce the extraction time, for example, to 1 hour instead of 24 hours or 72 hours. And also you can combine this with more quantitative methods, now the targeted analysis. So in that case, the compounds of interest, of concern that you have determined, for example, with your extractable study, can be followed up more precisely with a targeted analysis to have a more quantitative result on these compounds. Now, and with this, I can maybe finish with an example. So here you can see an example of an infusion bag with tubing, where we did an extractable study using water and 100% isopropanol at 50 degrees Celsius using exhaustive conditions. Now we did a complete chemical characterization study. So a screening for volatiles, semi-volatiles, non-volatiles, and metal. And as you can see here in the below, one plasticizer, the DHP, probably coming from a PVC component of the device, was detected above the toxicological acceptable level, with a margin of safety below 1. Now we did a follow-up study, a leachable study, a more simulated use study, where the bag was extracted at room temperature, using saline only for the entire shelf life. And the plasticizer, the DHP, was measured with a targeted analysis, so a calibration curve to have more quantitative results on the leachable amount. And here you can see the concentration for this plasticizer, in that case, was at an acceptable level, with a margin of safety above 1. So under normal conditions, the exposure to this plasticizer didn't present a toxicological risk to the intended population. So as you can see here, there are possibilities to address extractable studies with too many extractables at too high concentrations from a tox risk perspective. So with that, I want to end my presentation with a conclusion, some takeaway messages. So when doing a chemical characterization study, you have to perform an extractable study that represents the worst case chemical release to the patient. So here, the choice, your study design is critical. So the choice of your extraction conditions and solvents is crucial. Next, you need to calculate the AET that should be reached. And you need to report, identify, and quantify every compound above the AET. Now, a chemical characterization study is followed up by a toxicological risk assessment. If there are issues, for example, when you end up with too many unknowns, if the identification level is insufficient, you can move to second pass testing. So using high-end equipment to identify these unknowns. And if you end up with an extractable study with too many extractables at too high concentrations, there is the possibility to perform a leachable study, where you will simulate the real use conditions more. And you can combine this with a targeted analysis of the compounds of concern to have more quantitative results on these compounds, if there's a certain tox risk for these compounds. So, and I think this was my presentation. I want to thank you all for your attention. And if there are questions, I will try to answer them. Thank you.
Okay, thank you very much for that, Koen. And thank you as well, Alan, who started today's webinar for his presentation as well. Okay, great. So now it's time then to start